Good afternoon. Today is Sunday, May 31st, 2020. My name is Ricardo Sandoval, and this video is for week 3 of Biology 225 at Los Angeles Pacific University. This class is microbiology. This is lab number 5. This lab is entitled Bacterial, Morphology, and Staining Techniques. This lab will have 2 parts. In the first part, I will be taking 2 samples of my teeth and gums and then placing them onto slides using a stain and also heat fixing them, and then of course taking photographs and viewing them under a microscope. In the second exercise, I will be using previously cultured bacteria or microbacterium of E. coli, S. cerevisiae, and S. epidermis, and going through a staining process as well with that. So the first part, again, I'm going to collect 2 individual samples from my teeth and gums, and I did want to go over the supplies that I'll be using for this exercise. So let me flip the camera and we'll go from there. So I have my 2 sterile cotton swabs that I'll use to collect the samples from my teeth and gums. These are 6 slides. This is a stain called Congo Red that I'll be using. I also have a gram stain kit that contains safranin, crystal violet, a decolorizer, and then something called, looks like iodine here. I have a face mask that I'll be putting on shortly. A lens wipe just to clean the lens of the microscope when I need it, or the slides themselves. This tea light will be used to heat fix, or to basically heat the slide from underneath to fix the specimen. I have this to light my candle, and then I have a test tube clamp and a good old clothespin. Okay, so with that being said, I'm going to put on my gloves, put on my face mask, so I do apologize if I sound muffled, and then we'll go ahead and get started. And then of course I also do have my microscope that I'll be using to view the 2 teeth gum specimens that I take once they're stained. And then of course I also made this, which is just a quick little sign, just summarizing my name, today's date, the lab, class, school term. Alright? And then I will also be using this tray to stain the slides. Okay, so as I said earlier, I'm going to take 2 samples from my teeth and my gums. So I've gone ahead and labeled the first slide that I'm going to use, and hopefully you can see that it says 1 direct M. So the 1, of course, indicates this is the first sample or the first slide. Direct indicates that it's a direct sample or specimen coming from me, my teeth and gums, and M means it's coming from the mouth. The circle that you see is drawn on the underside of the slide, and it's just to give me a guide of where to smear the specimen on the, I guess, upper portion or the upright portion of the slide once I collect the specimen. I've already, just to preface this really quickly, cleaned my surface with my 10% bleach solution. So I'm now going to go ahead and remove this face mask and collect my specimen. So I'm just going to put this slide here on my paper towel. Just take this off for a minute here, and then pull one of these swabs out. Okay, and I'm just going to rub it here, like in a previous video. Okay, so now, let's put this back on. I have my slide here facing upright, and then I'm going to smear my sample in the center, just in the space that I created by drawing that circle. And I'm I'm just going to smear it in a circular motion, just to make sure that I have a good sample distribution here. Okay, so there's that. So I'm going to set this aside so I can dispose of it later. And now I do need to let this slide completely air dry. Alright, so it's a rather warm day here in Southern California, so my slide here has air dried. So the next thing I'm going to do is I'm going to heat fix the slide here, and using my tea light, what I'm going to do is I'm going to pass the slide over the tea light about 5 times. Over the tea light, over the flame here about 5 times, just to make sure that the sample's set. And it's just going to be a very quick pass for 5 times, because of course, glass and heats don't really do well. Because if I pass it too long, the slide will break. And I am holding it with this clothespin. Alright, so that's one pass. Two. Three. Four. And 5. Alright, so the slide has now been heat fixed, and I need to let the slide cool completely. Okay, so my slide has now cooled for a full 10 minutes. And the next thing I'm going to do is I'm going to stain the slide. And I'm going to stain it with a gram stain called Crystal Violet here. So what I'm going to do is I'm going to place 1 to 2 drops right on to my smear, which is the center of the black circle I wrote, and then let it sit for 60 seconds. Right. Okay, so one and 2 drops. Okay. So that's going to sit for 1 minute. Alright, so the Crystal Violet has sat for a minute. Now what I'm going to do is I'm going to rinse the stain off the slide using good old tap water. So I'm going to place the slide at an angle and then just rinse the slide until the color, if you will, from the slide is gone and my water runs clear. Okay, so that's done. So now I'm just going to grab a paper towel and just very gently dry my slide. So just blotting here, since I don't want any extra liquid on the slide, and then I'll show you here in just a second. Okay, that now in the center of the slide you can kind of see a small purple coloration, and those are my plaque cells that I got from my teeth and my gum line. Alright, so at this point with this first direct sample mouth slide, I'm going to take some pictures and upload it to my final report, and then we'll move on with the next slide and the next sample. Okay, so the next staining technique that I'm going to do is using a stain that is called Congo Red. This is a negative stain. So I've gone ahead and labeled the slide to -M, so second sample, negative slide, M per sample from my mouth. So what I'm going to do here is I'm going to place a sample of the Congo red here on the slide. Then I'm going to use a sterile swab and again collect a sample from my teeth and gums, agitate it in the Congo red, and then using a second slide here, I'm going to spread the stain across the slide. Then it will have to dry completely before I can view it under a microscope. Okay, so I'm going to go ahead and proceed with opening up the Congo red. Pair of scissors here. Okay. Okay, so there's a drop of the Congo red. Just going to set it to the side. So now I need my sample. So I need to remove my face mask for this. And again, this is going to go against my teeth and gums. Okay, so I have my plaque specimen here. So now I'm going to rub this or agitate this in the Congo red to spread the specimen. That's not what I wanted. Okay. So now I'm going to go ahead and this doesn't work out. Okay, so now using this other slide, I'm just going to my best here to do it at a 30-degree angle and then just draw the stain across the slide. Okay, so now this has to dry completely. Now after the slide dries, what will happen is I'll take a picture of it and then view it under the microscope and upload it to my final report, which is kind of difficult to show. So at this point, we're done with the Congo red portion of the experiment. Okay, so for the final portion of Lab 5, bacterial morphology and staining techniques, I'm going to use the cultures that I had previously cultured, the bacteria, I should say, of E. coli, S. epidermis, and S. cerevisiae quite a while ago. They've been sitting in my fridge since the last lab. So I'm going to place samples of those, one on each slide. The samples will then be heat fixed. They'll be stained with my entire gram staining kit, and then I will observe them underneath a microscope. So again, I'm going to flip the camera and just show what supplies I'll be using for this experiment. Okay, so again, I have my 3 slides there. This T-light is going to be used both to heat seal the specimens themselves and also to sterilize my bacteria tubes, if you will. The clothespin to hold the slides while I'm heat sealing them. My gram stain kits. These are the inoculation loops sitting in some alcohol to sterilize, and then I have my culture tubes here, E. coli, S. epidermis, and S. cerevisiae, that I will, of course, obtain samples from. So with that being said, I'm going to go ahead and don my PPE and get started. Okay, so I now have my 3 slides, E. coli, S. epidermis, and S. cerevisiae. So the procedure for transferring these specimens for each microbe or bacteria to the slide is going to be identical. So first thing I'm going to do here with, I'm going to start with E. coli, is I'm going to remove the lid from the tube. And then I'm going to flame the tube here, just to sterilize it. Make sure there's nothing living on it so I get a nice clean sample. Okay. So now I'm going to grab one of my inoculation loops that's been sitting in alcohol to sterilize. Just going to bang it here against the cup so that there's no alcohol. And it's nice and dry. Okay, so I'm going to place the inoculation loop straight down into my sample here, making sure not to touch the lid here. And then I'm going to flame the lid here of the tube once more before I seal it. Okay, so that's that. Lid's going back on, and then I'm going to set the E. coli tube to the side. So this is my E. coli slide. I'm going to press the loop just right in that circle I drew. And then just a couple of times. Make sure. Okay, now this is going to be set aside to dry. Just right here, and then off camera I do have some bleach where I'm going to soak this before I bring it away. Okay. So next, again, I'm going to do the same thing for my S. epidermis. So this is my S. epidermis tube. And interestingly enough, now that these tubes have, I guess, been incubating, if you will, for a while, although I don't know if incubation can happen in a cold fridge, so far the E. coli and S. epidermis do have a very distinct odor. Okay, so sterilized inoculation loop. Just kind of banging on the side of the cup here. Okay. So straight down into my sample. Just trying it for a second here. Straight back up. The lid of this, the tip of the tube gets flamed. Okay. Lid on, and then I'm going just again to set this aside. Alright. So my S. epidermis slide. Okay, and I'll just place the inoculation loop right on the circle I drew. Both sides, make sure I get a good deposit. This will be set aside to dry, and then again, this is going to go into my bleach down here. Okay, so my last culture here is my S. cerevisiae. This came from yeast. This is my S. cerevisiae slide. So lid off. And then I'm going to flame the lid, give me the tip of the tube here. Okay. My third and last inoculation loop. Just gonna bring the alcohol off. side. This is going to go straight down into my culture. Straight back out. Hold this and flame it. Okay. Lid back on. Set it aside. And again, this is my S. cerevisiae slide. I'm just going to place the loop here against this white circle. Or the circle I drew, not the white circle, this black circle I drew. Okay. And now all three have to completely dry before I proceed to the next step. Alright, so now all three of my slides for E. coli, S. epidermis, and S. cerevisiae have dried. So next thing I'm going to do is I'm going to heat fix them. That's the word I was looking for here. So here's my trusty clothespin that I used earlier. Let's place this on the slide. There we go. All right. So again, I'm going to pass this 5 times across the flame just for a few seconds. 1, 2, 3, 4, and 5. And I'll do the same thing for the other 2 slides. One, 2, 3, 4, and 5. And the last one here is for S. cerevisiae. 1, 2, 3, 4, and 5. All right, so now all 3 slides have to cool for 10 minutes, and the next step will be to steam them. All right, so all 3 slides have cooled for 10 minutes, and now I'm going to proceed with the staining process. So I'm going to use my entire gram stain kit, so it's going to be crystal violet first, followed by PVP iodine, a decolorizer, and then finally the saffron. Okay. So step 1, and what I have in my staining tray currently, this is my E. coli slide. So step 1, I'm going to place 2 drops of the crystal violet on the circle here where the specimen is. And then once I've placed the 2 drops on, I'll wait for 60 seconds and then rinse it with tap water. So there's 1 drop. 1, 2. And we'll wait just about a minute. All right, so it's been a minute, so I'm going to go ahead and rinse my stain again with tap water. Okay. Back into my tray. So next one is the decolorizer. This is 2 to 3 drops. Okay, that's 3 drops. No real viscosity to this, and we're going to wait 30 seconds. And then I'm going to rinse it again. Okay, it's been 30 seconds. Tap water and some rinse. Okay, and the last one is the saffron. 2 to 3 drops of this as well. One, 2, 3. We'll wait 30 seconds, and then this is my last stain. So this is the final stain. So after this, I will rinse the slide, allow it to dry, and then I'll repeat the entire process for the other 2. So 2 drops of the crystal violet, let it sit for a minute, rinse. 2 to 3 drops of the iodine, let it sit for a minute, rinse. 2 to 3 drops of the decolorizer, let it sit for 30 seconds, rinse, and then finally the saffron. All right, so it's been 30 seconds. I'm going to go ahead and rinse the saffron. Okay, so all of my 3 slides, the E. coli, S. epidermis, and S. cerevisiae, have been stained using the 4-step gram stain process. All 3 slides have dried, and the next step is for me to upload some photographs of the slides themselves, and then view them on a microscope and submit those photographs to see how the specimens show up best under a specific level of magnification. So at this point, the experimentation portion of this lab is completed. Thank you.